Section 21.1 and 21.2 goes over microevolution and the Hardy-Weinberg equation and how it applies to the evolution of populations. So the driving force behind change in a population is going to be microevolution, and this is the starting point for that change in an entire population. The name tells you what it means, evolution on a very small scale. The prefix micro tells you that. And what exactly it is, it's a change in allele frequencies in a population over generations. So a very small change is going to cause a big change that we see in the population. So frequency, how often we see something, or how frequent something is. And then to remind you, an allele is a particular gene that is in control of a particular trait. So there's an allele for flower color, an allele for, and it can be several alleles, but eye color, an allele for height, and so on. So again, alleles are genes, and how frequent we see them change is microevolution. Oh, and there's three driving forces behind microevolution. So we already know natural selection is the driving force always behind any kind of evolution, but genetic drift, which will be a event that will occur that can cause a change, and then gene flow, so transferring between populations. We'll get into the last two a little bit more in this chapter, but again, three things that can affect microevolution: natural selection, genetic drift, and gene flow. So the starting point in a population, and when there is going to be evolution, there has to be genetic variation. And way back, Darwin could not explain how inherited variations are maintained in populations. That traits, when you have two parents that are opposite of one another, why there isn't trait blending. So something like a white flower and a purple flower, like a deep purple, why aren't all the offspring a light purple, a blend of the two parents? A few years after Darwin's Origin of Species, Mendel proposed his hypothesis of inheritance, in which he addressed genes and alleles and how we have inheritance. So there are several sources of genetic variation. So again, having differences in genes in DNA in a given population. Mutations. Mutations are the only source of new genes and new alleles. So kind of like in the pocket mice example, it was assumed that all the mice had light-colored fur initially. And then there were these new genes, that black or gray fur. That would be something new within that population from a mutation, which causes in that entire population genetic differences. And something that's really important when we have mutations and we look at mutations, we're only concerned about the mutations that are happening in the gamete cells that are being passed to the offspring. So, at least in our body, with our trillions of cells, if we have mutations in our body cells, in our somatic cells, it doesn't really make a difference in terms of passing that on to future generations, because it's the gamete cells, the reproductive, the egg cells and sperm cells, when there's mutations there, that can introduce new genes and new alleles into a population. Something else are point mutations, which we've learned about that before. So a difference in a nucleotide being swapped out, so A's and T's and C's and G's. Chromosomal mutations, having duplicates, so gene duplication. Those are all examples of different kinds of mutations. Now, when we see these mutations and genetic variation, it affects prokaryotes, simple-celled organisms compared to eukaryotes very differently. And it has to do with how they reproduce. So in prokaryotes, they have a quick turnover. They reproduce very quickly. So often when there's mutations, it can quickly generate big genetic variation in a population because, again, they reproduce really fast. Within whether it's a human population, animals of any sort, with sexual reproduction, there is going to be genetic variation, but it's a lot slower of a process. And in this case, how it takes place, crossing over, independent assortment, and random fertilization. So to remind you, crossing over is when you have the chromosomes, the homologous chromosomes from mom and dad pairing up, and then they swap bits of chromosomes between them, and that's going to cause uniqueness when those gamete cells are eventually formed. Independent assortment, how the chromosomes, when they split off and during anaphase and they form new cells at the end of meiosis, that makes for unique egg and sperm cells. And then random fertilization. So if you've got millions of egg cells or millions of sperm cells, which ones actually end up becoming fertilized and turning into offspring? It's completely random. So all of those things lead to genetic variation. But again, comparing prokaryotes to eukaryotes, it's a very different process, whether it's quick or slow. But it does cause genes to be different in a population. But to say it one more time, the only way to get new genes and new completely different alleles is from random mutation. In order for evolution to occur, there has to be that genetic variation that I mentioned. But just because there is genetic variation occurring within a population, that isn't for sure that evolution is in fact going to occur. So in order to figure out whether evolution is occurring, has occurred, is going to occur, we need to learn a bit of information about the population. So what is a population defined as? It's a group of individuals of the same species that live in the same area at the same time that interbreed and they produce fertile offspring. And when we study populations and how they change genetically over time, that's what's known as population genetics. So we have to have an understanding about the population in order for us to figure out whether or not they are evolving. In order to understand a population's genetic makeup, we first describe its gene pool. Gene pool are all the alleles at all loci in all the members of a population. So first, what is a loci? So a loci is a segment on a chromosome, and it's a fixed position. So when we're comparing like this picture, looking at all the pigs, and you can see that they're all different colors, we're looking at a chromosome that is in control of their tone or their color. And it has to be the same location on the same chromosome in each of these pigs. So again, loci is a fixed position on a chromosome, and we try to figure out all the different alleles that are in that loci that can control, at least in this case, the pig color. So fixed allele, these would be all the members of a population that are homozygous for the same allele. So if when you're looking at this, and this picture is really silly, but if you're looking at the picture of all the different colored pigs, if they all had the letter A allele. So as a reminder, we use letters to indicate, and it's shorthand notation for alleles. So if we look at this gene pool and all of them were A's, then it would be a fixed allele, and they would all be the same color as well. And their gene pool would show less genetic diversity the more fixed alleles there are. So if, in now looking at the picture as is, you can see that we're showing you that there's A alleles, little a alleles. There's B's and C's and D's and E's and so on, in capital letters and lowercase. This picture is indicating that there are not fixed alleles, that there are different alleles. So in this case, this gene pool would have high genetic diversity, and you can see that again glancing at the picture with all the different color of pigs. The Hardy-Weinberg equilibrium describes a population that is not evolving. So when you have frequencies of alleles and genotypes, so genotype would be what you're looking at on the right, the C with the exponent basically R or superscript R, the C superscript W, and so on. Those are genotypes. So when those and the individual letters, the alleles in a population's gene pool, remain constant over generations, unless it's acted upon by agents other than sexual recombination. So again, if things are constant, that's an indicator that nothing is evolving, and that would be the Hardy-Weinberg equilibrium. There are rules or conditions for Hardy-Weinberg equilibrium in order for the alleles and the genes to stay constant. Some of these rules are there can be no mutations. So again, when you introduce a mutation into a population, that could cause new genes and new alleles, new genotypes, and then new phenotypes, which could have an advantage over other genes. So absolutely for equilibrium to stay in place, no mutations. Also, there must be random mating. So if we had sexual selection taking place, and again, whether it's with birds, maybe listening to a mating call, physical features, when you have organisms that are selecting a partner that's biased in a sense, that would cause favoritism of certain genes and certain phenotypes. And by having that take place, that could cause a change in that population. So random mating, or mating must be random in order for things to stay at equilibrium. There cannot be any natural selection. So you can't have certain traits have an advantage over others in a certain environment, because when that again takes place, and that really piggybacks off of the second one too, when that takes place, again, there won't be equilibrium. You also should have an extremely large population size. So when you have a really small population where there's variation in genes, and some sort of an event takes place which causes one gene to have an advantage, it's really going to affect that population because it's so small. And maybe that gene or that allele gets wiped out because of the event taking place and it being really devastating because the population's so small. So you must have an extremely large population size. So to define what genetic drift is, again, it's an event that takes place in a small population where the odds go up, basically, that some gene may disappear based on that event. Okay, so extremely large population size is important to maintain that equilibrium. You can't have gene flow. So gene flow is where you have genes coming in and alleles coming in from another population. So coming in would be immigration with an I, and then you can't have any members of that population leaving, which would be emigration, because that's going to affect the genes that are already there. So you can't have any of that for equilibrium to be maintained. So if any of those conditions are not met, then microevolution is going to occur. So again, no mutations, you have to have random mating, no natural selection, large population size is a must, and absolutely no gene flow. One of my favorite parts about this chapter and about evolution is using the Hardy-Weinberg equation. In understanding if a population is evolving, we need to understand the frequency of the alleles in that population. So there's going to be two equations, two Hardy-Weinberg equations we need to be familiar with. The first one, and the most simple equation, has to do with allele frequencies. So the equation is P plus Q equals 1. One represents either one population or 100%. So you can look at it either way. Now, what do the P and the Q represent? So the P represents a dominant allele. The Q represents a recessive allele. Now, typically speaking, we use different letters to represent the allele. So like capital A versus lowercase a. We could use uppercase B versus lowercase b. Whatever letter that we use in this equation, P is dominant, Q is recessive. So when we add up all the different dominant alleles that are present in the genotype of a population, and we add that to all the recessive alleles, well, those are the only kinds of traits that you can have, or genes that you can have, so it must equal one population, or 100% of the population. Now, at some point, we're going to need to solve for either P or Q. So just please note that basic math here, if you take 1 minus P, that's going to leave you with the frequency of Q, and then 1 minus Q will leave you with the P frequency. So please make sure you understand that P and Q represent the individual alleles, P for dominant and Q is recessive. The second Hardy-Weinberg equation that you need to familiarize with discusses the genotype frequencies. So again, I said this, I don't know how many slides back, you need to be familiar with the terms homozygous dominant, heterozygous, and homozygous recessive. Now, what those are? Well, they're being shown in the parentheses. Capital A is a genotype for some trait. When we see two capital letters, that is homozygous dominant. The prefix homo means the same, and in this case, they're capital, so it's dominant. When we see a uppercase allele and a lowercase, that's known as heterozygous. Again, a genotype representing some trait. And in both those cases, homozygous, dominant, heterozygous, those are both going to physically show the dominant trait. And the third genotype that we see being shown in parentheses is lowercase a. That's known as homozygous recessive. And in that, with that genotype, the organism is going to have the recessive trait, whatever that may be. Now for the equation. We have p squared plus 2pq plus q squared is going to again equal one population or 100% of some population. Now, what do all these letters mean? And it's a little bit similar to the last one talking about allele frequencies. When you have p squared, that means you have 2 of the dominant alleles, because p on the last slide stood for dominant allele. So if you square it, you have 2, capital A, homozygous dominant. When you have 2pq, that means you have a p, dominant allele, and you have a recessive allele. So in that case, and you have 2 alleles total. So in that case, it's going to be the heterozygous. Finally, q squared. Q represents recessive, so when you have 2 of them and it's squared, it is going to be, again, homozygous recessive. So now what does all this mean between the last slide and this slide? Again, these formulas really have to do with basic application and understanding the frequency of a certain trait in a given population. And what we use these for is once we figure out the frequencies of the homozygous dominant individual, the heterozygous, and those organisms in that population that are homozygous recessive, if we know the frequencies, and then a year later we figure out the frequencies, and then maybe a year later, on and on and on, we can see if the frequencies are changing. And in that case, that would be an indicator that evolution is occurring, and that would definitely go against that Hardy-Weinberg equilibrium if things are changing. So what do you need to know between the last one and this one? You absolutely must know what P and Q and P squared and Q squared and 2PQ, what all of those represent. And we'll do a practice problem in just a moment. Okay, hopefully you can see this. I just messaged out this picture, so hopefully you can see it and hopefully you understand it. So we're going to be applying the Hardy-Weinberg equations to a population, a made-up population of our class. So our class has about 26 students in it. And when we did the PTC paper, around 20 of us were tasters. And that leaves 6 of us being non-tasters. Now, what we know about the genotype of the tasters versus the non. We know non-tasters are homozygous recessive. So we use T's here, so it would be little t for their genotype. Because the only way to not taste that PTC is if you are homozygous recessive. So we absolutely know that genotype. Now, what makes it a little bit fuzzy is what is the genotype of the tasters? There's super tasters, which are homozygous dominant, and then you can also be a taster and be heterozygous. So again, you have either a capital T or a capital T lowercase t, and you'd still be a taster either way. So what we're going to figure out is the frequency of all the different types. How frequent do we see that recessive allele, lowercase t? How frequent do we see the dominant allele, capital T? How frequent is homozygous dominant? How frequent is heterozygous? How frequent is homozygous recessive? Okay, so what we know, again, are we know definitively the non-tasters. So 6 out of 26 is the frequency of the homozygous recessive. Something you must memorize. Q squared represents homozygous recessive. So going into the Hardy-Weinberg equilibrium, 6 out of 20 equals 0.3 frequency. Again, for those non-tasters. for homozygous recessive genotype. So 0.3 frequency is 30% of this population is homozygous recessive. Step 1, done. Now on to step 2. Once we know what the frequency of homozygous recessive is, we want to figure out how frequent is that recessive allele. So in order to figure that out, all you have to do is take the square root of q squared in order to get q. So I took the square root of 0.3, and that answer is 0.55 frequency. Again, what does that tell us? That 55% of this population has the recessive allele. Now it's important to note, wait a second, why did the frequency go up? Why is there a higher frequency, or 55%, compared to that 30%? Why is the recessive allele more frequent? So the answer is because when we have that homozygous recessive has 2 lowercase letters, has 2 recessive alleles. So right there, it's more frequent. The other part to this is the heterozygous genotype also secretly has that recessive allele. So it's almost like we're seeing that allele 3 times, and that's why the frequency increases. Okay, so now we have 30% of this population, again, is homozygous recessive. We know we see the recessive allele 55% of the time. Now what do we do? So now we have Q, the recessive allele frequency. From that, we can use the first Hardy-Weinberg equation. And that equation is P plus Q equals 1 population. So you rearrange it, 1 minus Q, which we just obtained from step 2, that's going to leave us with the P value. So when I subtract, I get 0.45 frequency. 45% of the population has a dominant allele. And again, P is what stands for the dominant allele. Okay, so now I have that. Step 4 and step 5, I do together. So if I have P, I have Q, I can figure out P squared in 2P squared. So now that I'm looking at this picture, I see I didn't put my work in, but hopefully it's self-explanatory. So I take 0.45 and I square it, and that gives me 0.20. I plug in my values for P and Q into that 2PQ, and that gets me 0.50. So again, what is that telling me? It says that in the population, 20% are homozygous dominant supertasters, and 50% of the population are heterozygous, and they're still tasters. So now I need to double check everything. And I double check using those Hardy-Weinberg equations. Does my P and my Q equal 1 population? Does P squared plus 2PQ plus Q squared equal 1? I hope so. So adding it up, 0.45 and 0.55 equals 1. Check, I did it right. I accounted for the frequency of the dominant allele and the recessive allele. Next step is the other one equal 1. 0.20, 20% supertasters, homozygous dominant, plus 0.50 heterozygotes, plus 0.30, that is my original homozygous recessive. Check, equals 1. I've done the entire 2 Hardy-Weinberg equations all the way. I know how many students, and I can apply it to the numbers. I didn't do that in this, but I can figure out how many students would be tasters that are homozygous dominant versus heterozygous. Please, I hopefully didn't talk too fast. I know I've been on the slide for like many minutes at this point, but this is a process that you must understand. Taking a population, Understanding that you're always starting with that homozygous recessive and you're working through the frequencies of the dominant trait, essentially. All right, here's the answers for the plant population example 2. Hopefully this example is correct. I just sent out a remind text message with the correct picture for the PTC. Even though my answers were incorrect, I really hope that you saw the process and know how to do it. I'm really mystified too when I checked my answer in the end for step 6, how on earth they actually calculated out to 1 when I had an error at the very beginning with my Q² value. But either way, here is what I hope is the right answer for example 2, and please make sure you understand the process of the PTC and then see my picture coming through via remind for the actual correct answers.